Hello.
Morning.
Morning. All right. So why don't we get started? Do you want to say something to start off the meeting, or?
Yeah, I'll have a bit of an update. Been working more on other things other than just computational microscopy.
Yeah.
So just give me a few minutes here.
Oh yeah, no problem. All right. All right. Why don't we start with talking about our meetings for this week? So we had 3 meetings. We had DevoWorm. Actually, I don't think we had a DevoWorm meeting this week. We had a DevoWorm meeting the previous week because we, of course, took last week off, and we had DevoWorm that last week. This week we did not have a meeting, and then we had Cognition Futures. And if Jesse arrives in the meeting, maybe he'll give an update on that. We were covering papers from that special issue of Royal Society A. So that is, just kind of going through those different papers. This last week we talked about the AI scientist paper. The week before we talked about another paper on reinforcement learning and TWIC states and things like that. And then, of course, we've had our open source meeting this last week. We didn't have one the previous week. So this is the DevoWorm meeting for June 29th. So we didn't have one in July 6th, but we did have one in June 29th. And this was where Barshan gave an update on his project. So this was, I think, a blog post that he put together on his training pipeline for et.al, which is his edge attention model that he's working on this summer. So he goes over that pipeline, talks about how you can use et.al for different problems in embryology. He did these smoke tests, and then he's done more, and I'll talk about it later in the open source meeting where he has some other advancements, but that was from that meeting. Then I presented a little bit on a project that I've been working on. This was something that I worked on with Dick Gordon and Hussein Ather for a book that's coming out. They're working on this project, or this larger project of cancer physics, and they've been really, they had up until the time that Dick passed away, they really made a lot of progress on it. There's a book coming out soon on cancer physics, on a lot of the different aspects of imaging tumors. And so this work kind of grew out of that work where, we basically want to use some sort of noise to mask tumors, to see if these methods can recover the tumor and reject the noise. But we want noise that replicates things that you would find in tissues. It's not enough simply to mask it with Gaussian noise. And so this was one type of noise that exists, and this is actually from the computer science literature, is this sort of procedurally generated noise, or Perlin noise. And so Perlin noise is something that you can simulate very easily, especially in something like Python. They use it widely in computer graphics and in video game design. And so we had been working on simulating Perlin noise and using it to embed tumors, and then being able to see if we can pull those out of the sample. So that's where this comes from. So one of the things in the paper, in the final version of that paper that was mentioned, was that we can create sort of tissue analogs from Perlin noise. And so let me go through here to see if I have an example of Perlin noise to show you. So what I did is I presented a notebook on some of these experiments that I did post paper. I actually did them quite recently because one of the last things Dick and I talked about was to pursue this work. He encouraged me to pursue this work. So this is an example of Perlin noise right here. And you can see that this is procedurally generated. So it's, you can tune the noise so that you can, that it's sort of tighter. And but this is an example of Perlin noise. And the idea is that this is, if we massage the technique a little bit, can serve as a tissue analog. It can serve to simulate the sort of the texture of tissue and the statistical properties of tissue. And so that's where this work comes from. So the first thing that in this notebook was to generate some Perlin noise, just some generic Perlin noise. And you end up with this two-dimensional array where it's generated over a number of steps, and you get this as a result. Now, one of the things about tissue, of course, is it's a three-dimensional environment, so this was expanded to the three-dimensional case. And then, we were able to simulate these viewers. We were able to view the three-dimensional volume of Perlin noise in different anatomical dimensions. So if you're going to be mimicking tissue, you need to mimic the way we image tissue. So we had these noise slices here, just experimenting with different ways to view that volume, which is not, this is not, a finished product by any means, but it just gives us ideas and ways to maybe to do this. So that's that part. And then this just kind of goes through simulating three different types of tissue. So there's fat, muscle, and trabecular bone. And those are tissues at very different specific densities and different textures. And then there are all these parameters that have to do with generating the Perlin noise, but also the different properties we might expect in tissue, like lacunarity, anisotropy, an angle, and then you're able to simulate those. And then these are some of the tissues that result. So you have fat, muscle, and trabecular bone. And then you have this fitness function that's being optimized. So there was, I think we're using a genetic algorithm here to select for tissues that were Perlin noise that look more like tissues. Okay, so this, and then of course we had this GA, and we replaced the GA with this, or actually we didn't replace the GA yet. We added this GLCM, or this gray level co-occurrence matrix. So GLCM is a texture method that quantifies texture analysis, analyzes pairs of pixel intensity values with specific spatial relationships. So we have the GLCM added to the model. We also added a spatial pooler to the model in lieu of the GLCM, and then compared the two. And then you end up with something like this, where you have fat, muscle, and trabecular bone. Textures are maybe need a little bit more work, but this is the difference in texture based on these different parameters. And then this is the Perlin noise input, the spatial pooler input, and then this SDR input, which is where the spatial pooler basically detects patterns in space. And then finally, there's this GAN. So, creating a GAN, which is a generative adversarial network, where you have a generator and a discriminator. And the idea there is to see if you can use a separate method to generate more refined tissues. And this was, I hadn't gotten this to run at the time of the meeting. So I still really haven't returned to it. So I'll probably be returning to that later. Okay, so Barshan had some comments. And then we went into this paper, could living cells use phase transitions to process information? And this was an interesting paper because they talk about cellular decision making and how cellular decision making is done through these phase transitions which involve these biomolecular structures called condensates. So there's been this recent interest in condensates as something that might have a biological function, condensates being large aggregations of biomolecules. So you might have large aggregates of proteins or peptides or something else in the cytoplasm, and the idea is that they serve maybe this computational function that then allows the cell to do different things to compute from its environmental inputs, to compute from some of the things going on in the cell that determine the state of the cell. So this just kind of, we just went through the paper, went through some of the graphs, and just showing these different aspects of computation. Okay, then, so that, actually, we had talked about that in a previous meeting, but I wanted to go through the figures in that paper in more detail, because it was important. I think it was an important paper in terms of thinking about, what cells maybe are doing in both internally and then in populations. Then we had this paper, Decoding the Architecture of Living Systems. It was kind of a counterpart to the last paper, and just talking about how people kind of think about biocomplexity. And then this paper, From Small Brains to Smart Machines. This is a nice paper because it shows how people are using the C. elegans connectome to actually address problems in AI. Using that in lieu of, say, like an attention mechanism or a neural network architecture. So the idea here is that, of course, that the C. elegans connectome, like all connectomes, don't look like a connectionist network. They are sort of connectionist networks in the sense that there's massive connectivity within the connectome. So if you have 300 cells, you have thousands of connections. And that's, kind of building upon that massive parallelism of the brain. And then also, we can translate that into sort of the massive parallelism of connectionism. But of course, the C. elegans connectome, like most connectomes, have other complex network properties. So they have this sort of small worldness aspect to them. They have the scale-free aspect to them. And they have this sort of ordered nature, or this ordered pathway through the network, so that you can end up with functional circuits. So it's not just random activity. You can really have some structured behaviors emerging. And in fact, in connectomes, you have things, circuits that evolve over time for specific functions. So that's what they're doing here. And they actually go through a number of studies where people have used replication of the C. elegans connectome to solve benchmark problems in machine learning and in AI. So they talk about a couple of these in the paper. First of all, they talk about sort of the nature of the C. elegans connectome and how that differs from artificial neural networks. And then they get into this aspect of these different models that people are using. And then they actually do these experiments where they're able to show, if you knock out certain cells at different points in larval development, you can get different results in the circuit. It's a really good paper. It does combine a lot of things that I think are of great interest to this set of topics on ANNs and BNNs and developmental AI and things like that. Okay, so I'm not going to cover the Cognition Futures meetings, but those are available on the JoePro YouTube channel, and so you can go there to see those. Like I said, we just covered some papers from the Royal Society, a special issue. This was our open source meeting from yesterday. So we didn't have an open source meeting on July 3, but we did have one yesterday. And so we started off by Barshan giving an update on his project et.al. This is where he was training the model. And so he talked about some of the things he was doing to train the model. And so he actually got into his data set, which is the EPIC data set, which is a C. elegans data set that involves, looking at different embryos, looking at their cell complements in embryonic development, and then looking at the cell tracking data and analyzing that. So he was actually looking at a number. So there are hundreds of embryos in this data set. Some of them are what we call wild type embryos, which are embryos where there's no modification of the genome in terms of defined mutants. They have a typical wild type genome, and so they don't have any defined mutations, as opposed to some of the defined mutants that you find in C. elegans research, where people will actually backcross to fix certain or to select for certain mutations in a strain, and then those mutations have some functional significance. And so he was working with these types of data. He was extracting the data, noticing that there was some data heterogeneity there, which we talked about. He also, wanted to be able to tune some of the hyperparameters for this graph that he's building from these embryos, and then, thinking about how effective this model is on the data. So he had some setbacks. He had some data heterogeneity, so he didn't have a balanced set of sample embryos for every case or for every condition. That's just something that's the nature of the data set he has to work with. A lot of our data sets are not optimized for machine learning. I know it's shocking, but it's true. But that's okay, because you can just work around that. But you just have to kind of be clever about it. And so then some of this had to do with the input data. Some of this had to do with, some aspects of the model itself. And so he just talked about debugging the model, dealing with a latency in the model, and then training this on CUDA, being able to do that. And then he actually showed this interface that was really interesting. He vibe coded this interface. Well, actually, he had some other things to talk about where he was doing some classification work on some of these subgraphs that he found. And he was looking at the attention weights. And he was just demonstrating, how you have, what the weight structure looks like across the C. elegans embryo. So what he's doing is building this embryo network where you have these cells in the embryo that are connected by distance. They can also be connected by signaling, or they can be connected by other types of criteria. What he's doing here is he's creating a graph based on distance for these cells, and he's looking at the weight, the attention weights, which are basically, have to do with their connectedness and how the attention mechanism is going to pay attention to these connections over other connections in the graph. And so he was going through this a little bit, showing how this works. And then he vibe coded this interface, which is really nice. I was complimenting him on it. It's basically a visualizer for this method. And so this is where we have the wild type, which is the control, or the perturbed embryos, which are where you have, they do these RNAi experiments to make sure that different genes are knocked out. This is the phenotype selection, so it's going to make a difference sometimes in what the graph looks like. And then he has this, just looking at the different cell-cell interactions and highlighting the connections that have a high level of graph attention. And it's kind of really a nice little interface, because he's able to kind of scroll through the embryo, look at the different cells, and then look at the different connections between this, or kind of think about the connections between the cells, that are the sort of where the attention mechanism is paying attention, and see how that's going to affect the final outcome. It's really interesting stuff there. So we talked about that for a while. That was good. He had some really, I think, really good, good solid first half of his GSOC coding period. So we're at the middle of the program, and so I think he's done a very good job of, implementing his project and getting some insights, some things that we can use for the group as a whole. And he's going to publish a blog post on this soon. Then Ashu gave an update on her work. Sarah came by and said hi to us. And then Morgan gave an update as well. Morgan actually has been doing some interesting things with his agentive coding. He'll talk about that in a little bit. But he has this repo that we kind of looked at here, BSCCM JAX. This is a computational microscopy workbench using the Berkeley single-cell computational microscopy data sets, and just kind of developing some tools for that on his own. Not forking any existing work, but just kind of drawing from what's been done and then building his own setup here. So this is just going through the repo and giving him a pat on the back for that. And then I had this topic that I cover every meeting for the open source meeting, and for this week I talked about this idea of different types of technologies and when to adopt those technologies. So this is an article on the bleeding, leading, dull, and rusting edges of technology. So the bleeding edge of technology are technologies that just emerge from some state of development. And a lot of times we'll adopt those kinds of technologies and not know what to do with them. And so this is a problem for a lot of organizations because they don't know how to use them properly, or they don't have procedures built in for them. But for other organizations, it's an opportunity to disrupt the market. Then there's the leading edge, which is where you're sort of an early adopter of technologies and you still have a problem with incorporating them into your workflows and things like that. But it's something that is contrasted with the dull edge of technology, which is where you adopt a technology that most people are using. And oftentimes you'll do this to keep up with other organizations. And then the rusting edge, where you have technologies that are obsolete or old and don't really have a lot of value other than maybe that they're legacy technologies. And so I actually presented 3 articles on this and just kind of went through these different aspects of different technology types, their sort of maturity, and then how that relates to innovation and other factors. And so this was a nice conversation. We didn't really have a lot of conversation per se on this. I kind of presented and we had a few comments at the end, but I think this will be something people I think will find useful. And going over it like this. And then we talked, of course, about how this relates to software design and relates to different project management strategies, such as DevOps and DevSecOps and Agile and so forth. So that was sort of the end of that meeting. So if you go to our YouTube channel, you'll find that we have some new content aside from our meetings. This is a presentation I'm giving at the Sandia XR conference this month. It's intelligence augmentation via ecopsychophysics and XR. And it goes over a number of topical areas. I talk about intelligence augmentation. I talk about different aspects of cybernetic control, some experiments in VR environments and AR environments that are very interesting with respect to these kind of theoretical topics. And then just building simulations around these type of eco-psychophysical phenomena. So that's what I'm doing here. So that's on our YouTube channel. I didn't really do it justice by just kind of going over it that quickly, but you can go and see for yourself. We also have a recap of our first 5 months of 2026, our different meetings. I usually do an AI recap. I do it in Gemini, and it generates a podcast format or a audio summary. And so you can go there and see what it's come up with. Usually has some interesting takes on the content of our meetings over that time. So what I like to do is try to digest these meetings by expanding out, the different arcs that we've been talking about over a 6-month period. So I can't remember what it came up with for this 6 months, but we've been talking a lot about agentive AI. We've been talking a lot about, I think we've been revisiting developmental AI, and we've been doing a lot of JoPro updates, so that's in there as well. Okay, Morgan, are you there?
Yeah.
Go ahead, give an update.
Sure, might have some more in a little bit. Sorry, the trains aren't running yet in San Francisco. So I'm just working out how best to get myself to the lab. Like I mentioned at I mean, you already covered the computational microscopy. This is like, it actually started as a because we've got a lot of open source projects, hardware projects in the lab that are focused on lab equipment. I've been just thinking about some of them. So there's a couple open source hardware microscopes, and the OpenFlexure is one of those. So this is a Raspberry Pi project that is just using a Raspberry Pi camera module, but it does get some nice features from that in terms of the, autofocus and some other nice things that are benefits for making a computational microscope, right? And so I was just starting to think about small variants of that would kind of not only be useful in trying to use this as a training microscope to teach people about optics, but but potentially to teach about some computational microscopy, where it's like, it's not just the microscope, but it's also about having a computer that can take a number of images and say something more from that, right? With some data processing. So that's how I kind of fell down the particular Berkeley single cell computational microscopy benchmark. The professor behind this, Laura Waller, is super interesting. Like has really been at the forefront of these kind of microscopy techniques. I thought it was also interesting that some of her postdocs, there in addition, she's also has a theme about teaching physics informed neural networks and what she calls physics learning, um, that comes from having these. She's a big JAX proponent, and so talks about how not only can you use this for um, um, uh, to insert a physics constraint in the loss function, but some additional ways in which you can bring this differentiability to bear on the problems in microscopy. Um, Fun fact that one of her postdocs who's led some of these kind of um, more physicsy discussions of computational microscopy is also the last author on one of the AGI ARC papers. So I think it's pretty cool when Francois Cholet is the first author and you're the last author. uh, uh, but um, so just really interesting group, and yeah, so that was one thread this week. The other being one of the other pieces of equipment that's used in the MIT How to Grow Almost Anything course, excuse me, that Elliot is a TA for, is the bioreactor. So this is something you use to grow cells, right? And in the synthetic biology world, you're growing cells typically because you've made some sort of modification to them. And what you're actually looking at is you're trying to get them to produce something um, that you want. So it's kind of like a biomanufacturing, although it can be some other things too. So a paper just came out called OpenEvo, and this is a directed evolution um, um, kind of direction that you can go with the same kinds of bioreactors, where you're growing something and then you're changing the environment, if you will, right? So one example would be like you're growing some sort of cells, you know let's just say yeast or something like that, and but you're also introducing salt, and you're changing the concentration of salt in the reactor. Or there's actually a version of this. I forget what it's called. It's like so these are called, they have a number of names. I think historically this was referred to as a chemostat.
Yeah.
I'll get a, I mean, you'll see if you go to, again, my repo, and GitHub M9H slash biopunk dash bioreactor. um, there's a paper, let's call it a white paper, um, and a set of slides that describe this, which with a bit of historical background, because yeah, so I'm blanking on the author of the chemostat, but sometimes these are called turbostats, and this relates to how they're being kind of monitored, I believe, in that they're using an optical signal in terms of kind of uh, how much the cells are absorbing light, to get a sense of the optical density. Um, but there's also a variant of this called morbistat, where what you're introducing is actually something like an antibiotic. So you're, introducing, if you will, poison, or something that's attacking the cells. So all of this kind of relates to people looking at growth curves of the cells, what are things that you can introduce to hold the cell population at a particular point, and things like that, right? Um, what you can see there is that there's a bit of uh, it's a great experimental platform for a lot of kind of microbiology. Um, so this is, you can see it as a kind of um, learning for um, biomanufacturing with kind of like a super practical angle. But you can also look at it as like a real educational platform for just um, how to do experiments with cells that you can learn a lot of different things from. And so the OpenEvo paper, which just came out in bioRxiv, um, is really um, the theme of what's called directed evolution. So again, there's some really cool historical stuff on this. Uh, I'm blanking on the particular um, is it the Latmer experiment? Um, is it who's the guy who's been doing the E. coli experiments for like 60,000 generations? Um,
Oh, Rich Linsky.
eh,
Yeah.
Lansky, thank you.
Yeah.
Yeah. So, and there's a four-letter acronym for that particular project. So it's the Lansky something.
LTEE. Yes,
Thank you.
LTEE. Yeah.
Thank you. Yeah. So it's again, synthetic biology has a bit of a biomanufacturing bias, I would say.
Right.
That drives it. Because it's like usually you're doing this to produce something that you want. Um, Although it certainly seems like in the great tradition of kind of knockout uh, as well, for kind of just learning about function, and so adding function is can be just as informative as taking away genes. So um, but there is that. Uh, a lot of these bioreactors, we get a lot of people come through the lab that are interested in, scaling up to much larger, um, uh, bioreactors, and a lot of what you're trying to do is learning about, optimal points in some sort of dimensional space, um, for highest yield and things like that, right? um, but I love the topic. I love yeah, and it's interesting, so just like with the microscope, these are kind of like $300 projects that you can put together yourself. These are open source. um, are there improvements that we can make? Are there changes that we can make where we can show people how to do kind of more complex experiments that are super useful for educational purposes? And so what the paper kind of ends with is you know like in this case, um, I'm comparing a bunch of different open source bioreactors for kind of what they're good for. And in the case of this particular paper, it's like what is kind of missing from it to make it into the platform that they actually talk about wanting to make. So their bioRxiv paper is like, hey, what we want to get to is this kind of AI controlled directed evolution. And um, and so I just talk about what that would need to additionally have that they don't cover in the paper. And it's kind of leaning on some other open source projects in terms of like, it'd be great to use, this feature from this project and this feature from this project, bring them together, and you can actually have a platform that they describe. So what I want to end with is um, like a nice, accessible, again, like hopefully this is still something that we can make for under $500. But in addition, we um, would be using CRISPR to show how kind of multifaceted CRISPR is in terms of reporting as well as modifying. Um, so, and how targeted CRISPR can produce a much more kind of, focal, if you will, directed evolution. Excuse me. Um, so that was the, yeah. So again, biopunk dash bioreactor. if if you want the complete AI generated paper. Um, uh, with the kind of history of this, as well as uh, ending with it's actually interesting to see how the I forget the PI at I mean, obviously Dudna is the main PI at Institute of Innovative Genomics Institute, but
Right.
like it was another person in her institute that kind of used these kinds of bioreactors to demonstrate principles of CRISPR. Um, and so I think this is going to be a really interesting, so what I'm really hoping for is that we're going to use the kind of fall where we're getting ready for the spring how to grow almost anything course, use the fall to demonstrate um, some new accessible experiments that demonstrate key um, key technologies of synthetic biology, but you know in a very how to grow almost anything kind of way, where it's like these are accessible educational projects. Yeah.
Yeah.
And then other than that, like I'm still working on neuroimaging and EEG and MRI processing is kind of my main thing. I've been doing some really interesting work in terms of trying to um, getting access to more data sets for kind of foundation model EEG work, MRI work with the MedArc AI people, um, and it was a great complexity meeting with the brain inspired group. They covered the well, they weren't really sure how to pronounce the person's name because it's like Raup, like RAUP. Um,
Oh, well, yeah.
like so, there was a couple of different pronunciations,
Hmm.
but just talking about that kind of that I think of it as a Darcy Thompson on growth and form kind of paper.
What was the
um,
paper?
it was the 66 wrote paper on a particular ammonite, or like
Okay.
a particular family, you know, um, and Yeah. So, Paul usually has someone do an intro, um, and the person was drawing not only on the 66 paper but also on the 67 paper. So I had not seen that. But I was also just, he had, he's Kevin Mitchell is a pretty regular um, attendee. So it can be nice to get his opinions on some of his takes on some of the papers.
Yeah, yeah.
Um, but this week it was he had Ricard Sol or Soleil. How do you,
Yeah,
I don't know
Soleil.
how to pronounce it. Soleil.
Ricard Soleil.
Um,
Yeah.
yeah, yeah. So that was kind of interesting. I don't think he really had too much to, I think more came from the person doing the intro, um, in terms of breaking down the paper. But it's what I loved was that he talked about how central he thinks his wet lab is, and his wet lab focuses on synthetic biology.
Yeah, right.
So I liked that, and he I think somebody was asking questions about his if I think they were saying, like, with, say, like ammonites or something like that, like, has anybody done knockout genetic work, right,
Yeah.
to look at that. And I think he was like, I'm sure they have. I don't think he knew if I don't think he could actually cite any particular papers. um but one of the things that he mentioned when trying to answer this question was that he likes having a synthetic biolab, and what he's really interested in is kind of um, modifying life. So doing something like, can I make microbes behave like ants or something like that.
Yeah.
And you know I loved that thought. I still don't know what that means, but I think that's super interesting.
I think maybe the collective
And
behavior aspects, probably.
yeah, yeah, I mean, I get what he, but it's just
Yeah.
what how would you begin to do such a thing,
Well, I mean,
like yeah, it's an interesting problem, right? But anyway, I did bring it up at the biopunk meeting because I was like, okay, we need to work on this. Like, I don't have any ideas, but I think this would be really cool. Um, uh, so that was Wednesday at the same time as Cognition Futures, unfortunately. You
oh,
know, I have raised this before, just to
yeah.
say, like I'm not sure this is a good time. But and then there's a great uh, that's also when I would normally meet with Neurotech X. Paris is like, more like 9:30, 9:40. But he usually rolls in a bit more like 10 a.m. This is the guy in Paris,
Yeah.
Makerspace in Paris. But there's actually another meeting that is weekly, that's focused on brain stimulation, that I attend now, um, which was this is a great group from, I think he's from Vienna, but doing TMS fMRI. So this is a great brain stimulation group seminar series. Um, Again, I've got some really interesting data sets to work with, and the everybody was really an ICML, so that's like the reason that a bunch of meetings didn't happen. Um, but probably why you're seeing so many um, ML results getting discussed is that people are uh, um, putting out their papers, or putting out their posters.
Yeah.
Um, the poster that broke the internet, at least on Hacker News, is it's the neuro AI group at it's neuro AI group at um, EPFL, and it's I'll see if I can pull the title, like the website's down because it got hacker newsed. Um, but it was about AI videos that maximally get a response at a particular brain area.
Oh, okay.
Um,
Yeah.
so I think this was causing a lot of consternation, at least in the Hacker News community, that this was, um, this is how the tech companies are going to destroy us. But um, this is actually somewhat in the vein of Andrea Tolias's work at Stanford, in non-human primates, which he calls inception, where he has neural networks designing visual stimuli to get maximally uh, responsive cell activity. Um, and it's really interesting what those visual stimuli look like, because they look really psychedelic, freaky. Um, so I think it's a really interesting area to study. Um, because like you can get um, kind of unnatural images that actually get maximal responses, right? And so you can call that, hacking your brain or something like that, like hijacking your brain's machinery or something like that. But I still, I do think that it's really informative in terms of how your neural networks are processing things in the same vein as um, optical illusions, right? Like it's the um, it still very much, tells you something about how these are connected and wired, and yeah. So I think that's interesting. I haven't seen too much. I mean, a lab at I mean, Jesse would know. Um, Bradley Wojtek, who's the chair of CogSci at UC San Diego, has a student who published something called Scale Surfer, which is a cool machine learning project, replicating or kind of like replacing a particular neuroimaging pipeline that we all use called FreeSurfer. And so FreeSurfer runs in like 6 to 8 hours per subject. Um, there's a Fast Surfer project that I've been using for a couple of years now that does it in a couple minutes, and Scale Surfer, which was also I think timed with ICML, does it in under 2 seconds. So and is doing something that's very interesting in its multi-scale nature. Anyway, because I've got so many data sets ready, it was interesting to run that, be able to give him feedback in terms of like, it is super interesting and it is damn fast. But it's got some accuracy issues, as as you can imagine, for
Mhm.
2 seconds.
Yeah.
Anyway, lots of updates, and I'm not going into the neuroimaging stuff. Um, oh, and then yeah, we've got our paper for the next CogSci reading group in SF, our in-person CogSci reading group, which is this Blazy Agaris. Um, no, I'm not pronouncing his name right. But it's an interesting paper about LLMs as communities and how they fine tune differently in communities, and it made me think a lot of you know um, the open source community work, or the sustainability work, in that it had groups of LLMs interacting, and some interesting things that happen with those. That I'll try and post that paper because I think it'd be really interesting to talk about. It's the reason. It's like something about it's like his January. If you pull up his Google Scholar, it's like a January paper from uh, that's like something about reasoning thought, um, differences from community.
Who is this person?
Blazy Agaris.
Oh,
He's
okay.
the Google VP.
Or Agera Arcus. Okay, yeah.
Yeah.
Reasoning models generate societies of thought.
Yes. Thank you.
Okay, there we go. All
Perfect.
right.
Yeah.
So yeah,
Yeah.
let's.
Yeah. I think it's interesting. Um, and I was saying to the guy who runs the meeting that, like, he was kind of apologizing because he used to work for him, and um, like as if he was maybe biased or something like that. But I was just like, no, I think I think it's a really interesting, um, something that people don't talk about, or like are not usually discussing, because they're not usually talking about LLMs kind of in communication with each other. So
Yeah, yeah. So I have
I
some. Oh, go ahead.
no, that's great. I'm gonna jump on the train now. That
Okay.
trains start running at 8.
All right. Yeah, so let's go over some of these things that you talked about. I have them pulled up. I'll go through them one at a time. So
Cool.
this is the paper you just talked about. Reasoning models generate societies of thought. This is on the arXiv. It's Blaise Gary Urkus, James Evans, Nino Scherer, and Junsol Kim. And so this is thinking about large language models and about reasoning reinforced models, including OpenAI's O series and DeepSeek's R1, outperform other merely instruction tuned models on complex cognitive tasks, are attributed to extended test time computation through longer chains of thought. So talked about chains of thought or reasoning chains. We've talked about that in the past. And this is something that, it goes sort of at the back end of the large language model pipeline. So here we show that enhanced reasoning emerges not from extended computation alone, but from the systematic simulation of complex multi-agent interactions. So this is the society of thought that they're getting at here. So you have this multi-agent environment, or this agent-based environment, where the agents are interacting, and that's where you're getting this power from. And this kind of goes back to the classic book Society of Mind, where the idea that um, you know minds are social as well as just sort of computational. And so this is where they think about this idea of having an agent-based model of reasoning. So this sort of model enables a deliberate diversification and debate amongst internal cognitive perspectives, characterized by distinct personality traits and domain expertise. And so that's, you know, kind of as the models are heterogeneous and they're chatting, they kind of come up with their own sort of chain of reasoning. And then, yeah, I think that does relate to what we're doing in, you know, open source sustainability, because you do have this agent-based layer to the model. And the idea is that you want to, well, I think in open source or in open source communities, you have this inherently social aspect of them. So, you know, you have to have individuals working together on certain problems. You can do things kind of
Right.
on your own, but you really do need to have that interaction to see where, problems get solved.
Yeah, yeah.
And yeah,
I think it's yeah. I mean, I think the difference here is that they're kind of like allowing the models to fine tune while in this kind of community setting. You know that they're anyway. But go ahead. Yeah, but I do think there's overlap. All right, they might be interesting.
through quantitative analysis using classified outputs and mechanistic interpretability methods applied to reasoning traces, we find that reasoning models like DeepSeek R1 exhibit much greater perspective diversity than baseline models, activating broader and more conflict between heterogeneous personality and expertise-related features during reasoning. This multi-agent structure manifests in conversational behaviors, including question-answering sequences, perspective shifts, and reconciliation of conflicting views, as well as in socio-emotional roles that characterize back-and-forth conversation, which together account for over 60% of the accuracy advantage in reasoning tasks through both direct and indirect facilitation of cognitive strategies. So this is, where you have this conversational agent aspect. It's not just, a person conversing with the agent, it's the agents conversing with one another in the context of that problem. Controlled reinforcement learning experiments further reveal that priming models of conversational scaffolding, even when dialogues lead to incorrect solutions, substantially accelerates reasoning improvement compared to answer-only training. These findings indicate that the social organization of thought, rather than correctness alone, enables effective exploration of solution spaces. We suggest that reasoning models establish a computational parallel to collective intelligence in human groups, where diversity enables superior problem solving when systematically structured and suggests new opportunities for ancient organization to harness the wisdom of crowds. Yeah, that's definitely in line with the spirit, at least, of agent-based modeling and sort of what you achieve with that. Yeah, it's interesting to see that they're thinking of sort of this wisdom of crowds idea. So that's what they're getting at, almost like crowdsourcing amongst the agents. This is the Hacker News article on this, or the Hacker News post on this article. AI generated videos to maximally drive a target brain region. So this is where, and then this is, as you mentioned, there's a lot of commentary here.
Sometimes, sometimes Hacker News is all for the comments, not
Yeah.
not the link. Yeah.
Yeah, so I mean, it's, let's see if they have the actual paper. This is the paper here, where
They did,
they
yeah.
advertise
Okay,
the
so it
paper.
comes up now.
Yeah. This is
Yeah.
NEVO, Neural Guided Evolutionary Video Synthesis for Dynamic Visual Selectivity. So they're using some sort of neural evolution approach, are they?
Well, yeah, but like, so what's their? Because like, I don't think they generated data. I'm kind of afraid that they used something like Tribe, and so they've got another model that's actually giving them a a sense of the neural activation. But yeah, you can check because the website was down last night. Yes, yesterday.
Yeah, yeah. So this is the arXiv paper, the Hugging Face code, and then this is, I guess, more Hugging Face code. I don't know why they have two links to it, but so the question that they start up here with is, what kind of video would make a given part of your visual brain light up the most? I guess they mean visual cortex or visual areas in cortex. And then this means the highest activation, or what would give an activation in a certain area. Nevo answers this in silico. So this is, I guess, a simulation. It involves AI-generated videos to maximally drive a target brain region, then uses them to map how visual selectivity becomes increasingly dynamic and social along the lateral stream. So this is the lateral stream. Um, you have parietal areas, occipital areas, and that whole stream of the visual cortex. So these are their standard ROIs: V3A, MT, I think, I don't know what that is, PSTS, PPA, PFA, ASTS. So these are all kind of the areas of interest. And then they have the natural images. So I guess these are presented to the model, and then the model outputs activity, and they see what the activity is. And yeah, I don't know if they're actually using real data for this or if they're just simulating the data. It's hard to say right now. You'd have to get into the paper. Searchlight
Yeah.
regions. Oh, go ahead.
No, I was just gonna say I'll definitely check as soon as I get into the lab. But
Yeah.
I have this sneaking suspicion that they're using Tribe.
Okay. Yeah, so this is searchlight regions, a dense searchlight across the surface. Each covered sphere has its own synthesized stimuli. So this, I guess, these are just kind of different locations in that pathway, and these are the areas that respond to these images, if I'm not mistaken. So how Nevo works. The first is you start with a digital twin of the brain. So this is, again, just saying that they're simulating it. We train an encoding model with digital twin that predicts how each visual region responds to any video. Now we can ask which video would make a chosen region light up the most. Nevo searches for that video automatically, using the twin's prediction as its reward. So I guess, okay, so we have this evolutionary prompt search, and then we have the prompt, which goes to the video generator, then the visual stimulation, which goes to, I guess, the digital twin, which I guess is based on human brain data, but is a digital twin, and then a reward. Whatever is stimulated in the visual digital twin is fed back to Nevo as the reward. And then there's another prompt to the video generator, and so on and so forth. So this is again, yeah, it is sort of, I don't know if you could say, like, it's based on, it's trained on data, and there's a digital twin that then is the thing that's activating. Yeah, that may be what they're doing. Okay, and then step two is evolve the prompt. So every video is described by a handful of genes, which are just like a genetic encoding where you're encoding different properties of the scene. So subject, lighting, motion, mood. Nevo treats them like a population, which just means they generate a population of these genomes, generate a batch, score each with the digital twin, then keep, mix, which would be done by a crossover operator, and tweak or mutate the best outcomes. Over many generations, predicted activation climbs. So I guess you're basically encoding these images of different properties. You're seeing how they activate the digital twin model. You're rewarding, you're basically selecting for those images, and then or those properties of images, and you're reinforcing them by generating more similar images. I guess that's how they're doing this.
Yeah.
Do you have
I,
something
I,
you
I,
want to add?
well, just like these models are kind of designed. I mean, again, I'll dig in when I am actually in front of a proper computer, but um, these models are better at giving you the spatial location for this than they are for, really predicting activity in the Andreas Tolius kind of way, where,
Yeah,
he's got like neuropixels in actual
yeah,
non-human primates. But I could be wrong. But it's interesting, and it definitely says something about the kind of response that it got on Hacker News about
yeah, yeah.
how people feel about this.
Well, I mean, I understand when you're doing it. I'm not really sure, because people have done this before with like, um, there've been like applications where people have taken a bunch of images and selected them by human input. Like humans would like say this looks nice, this doesn't look nice, and then they would evolve a population of images that were, I guess, aesthetically pleasing or something like that. So you're doing basically the same thing. You're using the selection criterion based on this digital twin model. You're creating basically images with these properties. You're you're recombining them and mutating them in different ways, and then testing them against the digital twin model. But it's like I don't understand like if it's just taking specific parts of the pathway and using that as sort of the reward or what exactly the reward structure is. Um, but anyways, yeah, I mean it's definitely in that vein. I think they're trying to sell it as sort of an updated sort of AI buzzword version of this typical closed loop feedback of like someone uses evolutionary algorithm to select things.
Yeah,
Yeah,
yeah, yeah. It's just that, like, whether these quote digital twins in neuroimaging are sufficiently predictive, um, is the digital twin is doing all the heavy lifting in this particular scenario, and I don't think it's really ready for it. But that's a separate
yeah,
issue.
yeah. I mean, you like, um, there is this whole debate in neuroscience. I don't know what the state of it is, but like where there's certain cell, like the grandmother cell idea, where certain cells are tuned to certain types of images. Like if you have an image of a famous person or someone you're very familiar with, and that cell or that part of the brain is active when you see a picture of that person. And at first, when I saw this, I thought, well, they're just seeing where these images are encoded in the model. But I don't know if that's true. I think it's just kind of like whatever is activated. So yeah, I don't know what how the digital twin is actually behaving. If it's at like a full scale representation of the images, or if it's just responding to different properties of the images, or what. It's probably worth thinking about. But so yeah, then there's number three, which is just about like these mapping this to video and an image video model. And then number four, just custom videos for every region, so you can do customization of videos um, and then okay, this is up the lateral stream vision turns social. So as you move up the visual stream, um, you end up with these sort of social things. So you have this social response as well as just simply the image response to image properties. Then probing with abstract stimuli. So then there's some experiments they ran where they tested different types of abstract objects and like these abstract stacked disks which have faces and how that activates different parts of this visual stream or this visual pathway. So, the digital twin includes these different visual components of the visual pathway as they're functional in the human brain or in human neocortex. And so, I guess the idea is that they respond in a like way. And then you can probe these different parts of the visual pathway with different types of stimuli. Okay. This is a YouTube video.
This is Andreas Tollius talking about the kind of real experimental work that is actually happening here.
Okay. Okay. Yeah. This is a paper by Andreas Tollius et al. Nature Neuroscience. Inception loops discover what excites neurons most using deep predictive models. So this is, I guess, an inception loop is like an analogy of the movie Inception. So this is finding sensory stimuli that drives neurons optimally, essential to understanding information processing in the brain. However, optimizing sensory input is difficult due to the predominantly nonlinear nature of sensory processing and high dimensionality of the input. We developed inception loops, which are closed-loop experimental paradigm combining in vivo recordings from thousands of neurons with in silico nonlinear response models. So this is kind of this in silico aspect of the brain modeling. Our end-to-end trained deep learning-based model predicted thousands of neural responses to arbitrary new natural inputs with high accuracy and was used to synthesize optimal stimuli and most exciting inputs, or MEIs. For most primary visual cortex, MEIs exhibited complex spatial features that occur frequently in natural scenes, but deviated strikingly from the common notion that Gabor-like stimuli are optimal for V1. So this is where they're actually probing the brain in different ways, where they're developing things based on recordings, and they're actually showing that there are things that maybe aren't in accordance with commonly held wisdom. And this point about V1 is where in a lot of experimental settings they'll use like a Gabor filter to generate an object, and those are the kind of stimuli, like these striped or modeled objects, very simple primitives, and that's what V1 is doing. It's distinguishing those kinds of objects. And what they're saying here is that in natural scenes, you have this deviation from that. When presented back to the same neurons in vivo, MEIs drove responses significantly better than controlled stimuli, or than the control stimuli. Inception loops represent a widely applicable technique for dissecting the neural mechanisms of sensation. That's so that's kind of a similar thing to what they're doing in the NeuroSim poster. So this is a paper that kind of it's not the paper that they talked about in the reading group, but this is a paper that is on this idea of ammonite form and this orthomechanical basis of ammonite form. So this is Alan Gorielli and colleagues. So it's mathematical biology. This talks about ammonites. Ammonites are a group of extinct cephalopods that garner tremendous interest over a range of scientific fields and has been a paradigm for biochronology, paleobiology, and evolutionary theories. Their defining feature is the spiral geometry and ribbing pattern, through which paleontologists infer phylogenetic relationships and evolutionary trends. So here they develop a mathematical model or a morphomechanical model, more specifically for ammonite morphogenesis. While a wealth of observations have been compiled in ammonite form and several functional interpretations may be found, this study presents the first quantitative model to explain rib formation in the ammonites. Our approach, based on fundamental principles of growth mechanics, give a natural explanation for the morphogenesis and diversity of ribs, uncovers intrinsic laws linking ribbing and shell geometry, and provides new opportunities to interpret ammonites and other molluscan evolution. So this is just going over this area. So like Morgan said, it's very much like um, a Darcy Thompson type thing, where Darcy Thompson, of course, was looking at like shelled animals and looking at the mathematical properties of their growth and their shape, and then plotting those out in different ways. And so it's very much, I wouldn't say it's a lost art, but it's not something that we usually talk about. I mean, people are interested in genes and they're interested in sort of building computational models of, you know, different molecular assays. And then this area has just kind of been like kind of a curiosity. It's nice to see that people are focusing a little bit on this and developing mathematical models to describe the phenotype without really reference to anything else. It's just kind of an interesting problem space. And so their morphomechanical model is here, where they show this image. These are the molluscans that they're talking about, the ammonites. You have this layered phenotype here, where you have the periostracum, the shell, and the mantle with different lobes. And this is sort of a cross section of these fossils here. You have this model where they show the periostracum radius, the mantle, which is the outer lobe radius, and the stress-free state, the everted state, which is a difference, change of state, and then this MEP biring, which is this combination of the two states. And then they show this cross-section here of the shell, where there's this generative zone, so it's showing growth in the shell. So as the shell expands outward, it grows using, through these rings, where you have the calcified shell and this generative zone. It's basically, these are what they call the ribs, these different segments. And so the rib growth is shown here, and they build a mathematical model of it. Okay. Morgan mentioned the long-term evolution experiment. This is the long-term evolution experiment, the site for it. Rich Lenski ran it for, since 1988. I think he's recently handed the project off. And so he's now, I think, retired or semi-retired.
Yes,
But they've been
he
running
passed
this.
it on.
Yeah,
Yes.
yeah. So this is, it started in 1988 and continued into the 2020s. It's now, I think it's 75,000 generations at least. And so that this is where they've, every day since 1988, they've passaged a culture of, or multiple cultures of E. coli, multiple strains. They've been doing this since 1988. So the idea is you have this continuous population of E. coli. You have them frozen down, and you can, freeze them down, revive them, but you always have like this core population going at all times. And the idea is you can use these evolved populations to test hypotheses in evolution. Since you have so many generations, at that point you can see different things evolve, different types of changes in the cultures and so forth. So, it's a very controlled environment for evolution. They control the inputs, they control the transfer process and the nutrients in the culture. And so, yeah, this is a little bit about, sort of the technical details and, just thinking about how long they'd run this experiment. So we're already at 75,000 generations, which is pretty good. A lot of papers have come out of this project, and E. coli actually are a good, people have run experimental evolution with E. coli, with yeast, even with mouse populations. And so this is an excellent paradigm to sort of understand the effects of evolution, in a way that you can't necessarily do, say, with fossils or with computer simulations, because you actually see these changes happen over the course of the experiment, and you can actually then, analyze the data and see the dynamics in front of you instead of having to rely on inference. All right. This is OpenEvo. This is the first paper Morgan talked about. This is Sebastian Cocioba, who I follow on Blue Sky, Alex Bisson, and some other people, familiar names. And they've worked on this open source platform for automated evolution and analysis. So here they introduce OpenEvo, a fully open source, low cost turbidostat platform for automated continuous culture and directed evolution experiments. So directed evolution is where you have this, experimental environment where you can, evolve the population in different directions. You can give it different selection pressures and so forth. And just automating this process is important, because if you're working in a highly controlled environment, you have to maintain that control in a way that isn't disruptive or confounding to the experiment. Now, their point in this paper is they want to have a low-cost platform. So existing tools are expensive, complex, or lack open source hardware. OpenEvo addresses this gap with a complete, fully automated evolution platform with detailed, illustrated construction instructions for beginners, open source software and firmware, and priced around $300. So yeah, their focus is on a very low cost platform. I know Sebastian works on, like, out of his garage or something like that, and is really kind of an expert at developing these kind of tools for low cost scientific endeavors. So this is always
Okay, that's interesting. That's good to know.
yeah.
Cool.
Yeah, he did.
Do you know where he's based?
I think in Long Island in New York, I believe.
Okay. Cool.
Yeah. So it's good to have someone who's kind of like living that lifestyle be able to develop something like this. And then so this is great. You can have a very affordable system where you can control the environment of your experiments. And of course, like Morgan said, this is something that you can do in things like the E. coli culture. You can do things in other types of organismal cultures. You can even do it with C. elegans if you wanted to, because you have, you might want to control those kinds of environments. Although I think their focus is more on these bioreactor type environments. And so, yeah, keeping a bioreactor stable is important. And having these kind of automated systems it's not easy to do, actually, especially at a low cost, because you need to have different controllers and other things, and you have to know how to configure it. So this is great. An optional PC-based interface offers enhanced functionality, including remote access, programmable evolution cycles, programmable LED stimulation, and a data visualization tool. OpenEvo can cycle through 3 types of media for positive, negative, and neutral selection conditions, supporting a wide range of experimental designs. We validate the use of OpenEvo by evolving Halopharax vulcani to grow from 15% to 12% salt over 150 cycles at around 1,000 hours, or in around 1,000 hours. Evolved cells grew 55% faster than wild-type cells at 12% salt, so that the salt concentration was varied, and they were able to basically see where they could get improvements in performance, but also see sort of the resolution of the approach. So if you vary salt, just kind of in a way that's not controlled, that's easy enough to do, but then does it actually show the growth changes that you would want? Whole genome sequencing of adapted cells found SNPs and large deletions. We also demonstrate positive and negative selection using the OpenEvo LEDs, a derived optogenetics via a phytochrome B-based optogenetic tool, with light as a selection stimulus during over 4,000 hours of growth. So they can actually incorporate uh, 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 optogenetic tools into this platform. And so that's actually pretty excellent, because it's going to be, it allows you to integrate all aspects of your experimental platform. OpenEvo lowers the technical and cost barriers for continuous evolution experiments, serves as a teaching tool, and is designed to grow an open community of users who share modifications. Excellent. Yeah. Did you have anything else to say about that, Morgan?
No, that's I mean, that's definitely the paper that kicked off this particular kind of, research crawl, if you will.
Yeah.
Sorry, there's a lot of along the streets of San Francisco. So anyway, okay, the ambulance is going the other way, but So what came up was like some discussion on Twitter about whether the the fire engine is following me. give me 2 minutes because it's coming right by. Okay, is fast. So one, I kind of wonder how they did their research for this particular paper, and I'm suspecting that they might have used LLMs.
Yeah.
because they missed a lot, and some of them are kind of like mistakes that I think are mistakes that LLMs make. But I still think it's a really interesting paper in terms of. Sorry, our front door has a new. Our front door has a new panel. 1 second.
Oh, okay.
It's asking me to open up my app. It's usually just touch it.
Oh.
So I'm gonna have to figure out how to do this. Okay. So yeah, it's so they missed some other projects that are absolutely in the same area. And some people on Twitter had kind of called them out for
Okay.
that. They also, but that I thought his response to that was very good in the sense of like, this is why it's a preprint. And,
Yeah.
this is why preprints are out there so that we can find these things. And but they also got some things wrong about some of the open source projects in terms of what their capabilities were and like which parts of them were open source. And that was the bit where it was just like, okay, I think maybe there's some holes in this paper that that I can fill, and I was already interested in terms of looking at what kind of like what was on offer, or what were projects that we could incorporate into educational curricula
Yeah.
that
Yeah.
would be yeah.
I think the educational
Go ahead.
part is pretty nice. Just thinking about how you would use that for, students, because you don't want to have really high-end equipment for students. Usually in teaching labs, they have like the base equipment or stuff that's old and used. but you know, and then allowing students to like modify things too, because you don't want
Yeah.
to have you know, things set up at a certain under certain parameters, and then you know things kind of go bad when you try to demonstrate something.
Yeah, absolutely. I mean, it's just to some extent, right? I mean, it's just like there's definitely that. In this case, um, the affordability is nice because, you know, in the same sense of like you can now afford to give everybody a Bunsen burner kind of thing,
Yeah.
right? Like now, you know, like you don't have to get everybody to use the same equipment,
Right.
in that sense. Here, I can switch over now. Yeah, I'll just stick with the Apple mic for now,
Yeah.
because this is a Linux workstation. But yeah, everybody can run their own project with it, you know, because it's so inexpensive, and potentially, you know, you can make equipment that's cheap enough where now somebody can modify it for their experiment, and it not be a big deal, because it's like if you want another one of those, then you know you can build another one. um so yeah, and like what the um what the paper for the biopunk-bioreactor is all about is like how we can extend OpenEvo for the things it's missing, and kind of learn from the other projects that are kind of in this space, which there are a lot, um, uh, to like get good design ideas and kind of make a best of breeds version that incorporates the um, all the best features into, you know, this kind of self-contained platform that, you know, allows people to do these really interesting uh, biology experiments, you know. So it's yeah, and they use a lot of different technologies that come together to um, yeah. And so what I'm particularly happy with is again, the paper is good.
Yeah.
In you know, collecting a lot of information from this particular field in terms of both the history of this, in terms of how much good biological theory has been experimentally derived from these kinds of bioreactors, as well as how the very development of CRISPR comes from these kinds of um, this kind of experimental platform. And
Yeah.
so, you know, like again, like it's we're as a kind of community science lab, um, to be able to demonstrate CRISPR in this kind of experimental way would be such a terrific uh, uh, you know,
Advance.
educational experience. Yeah.
Yeah. Yeah. Yeah. That's great. Yeah. So then, the final thing I wanted to bring up from this was this is the repo, Morgan's
Yeah.
repo,
Okay.
biopunk-bioreactor. So
Yeah, and you can see the manuscript. So there's a couple folders there.
with manuscript and slides and posters. So if you go to the PDF there, obviously, like I don't hide the fact this is AI generated, but again, I have a lot of not guardrails on it, but I put in a lot of effort into making these really information rich. And so it's both a comparison of bioreactors as well as a critique of the OpenEvo paper, which ends. If you go to the slides as opposed to the poster,
Okay.
I think the slides are a bit better in terms of So the benefits of these, what they're used for in terms of kind of the roles of this equipment in a particular lab, and and then some of our kind of like assessment of the pros and cons of each. And as you go down, you'll see more about just the what were some of the history of these in terms of how they kind of what they came from in terms of the chemostats. You can see there, those are the seminal papers for I think it's the Novick papers, and of course Minot. But that would be like 48, and I think the Novick's like 50. But again, it's like they have a long history. There was a bunch of papers, kind of like 20. I mean, certainly, again, long history. But the like 2018 to 2022, there was just a bunch of papers where they were really being used for experiments as well as being demonstrated as a kind of open source hardware technology. So you've got both like people coming out with this really inexpensive and accessible tool, as well as people demonstrating something very cool with it, or building on top of it, which is, again, the nice thing to see is like when another group takes that and then uses it to do some real science. So that's and so then I just try and go through some of the particular technologies of it, and and we make some recommendations. You have to hit the more pages at the bottom there to see the more slides. Yeah. But so there's some just trying to point out what is missing from it, and kind of like how we can put together something which again is like kind of leveraging the best of each project, to make something that again is like an accessible tool that allows you to do frontier science.
Yeah, I like this. And then I like your kind of distinction here: proto, devo, and bioreactor, the evolution, and the proto, sort of the characterization.
The Devo is confusing because, of course, it makes me think of dev, but of course it's directed evolution.
This seems to be a bottleneck in terms of automation. So I know, like, it takes, depending on your organism, it's going to take a while to do, and of course you need to have very precise be able to apply the same transfer techniques generation after generation, which is of course something that automation would be good at. But on the other hand, getting the data out and recording it at every generation, is that a problem in terms of automation, or is it just that no one's really done that yet?
it's you know, their point is it's easy. Their point is how to sample the system accurately while it's still cooking, so to speak.
Yeah,
Right.
yeah.
And
Right.
and you know, so like you'll see in this, it's got there's I mean, one of the biggest problems that we have is, of course, contamination.
Right.
And you know, getting so any time you can have a closed system that you know precludes those kinds of issues is a benefit, right?
Yeah.
The problem becomes how to also really sample the system. And you know, so again, there's some small discussion around, you know, how light has typically been used to kind of assess the system. Obviously, you'd like to do more than that. Um, um, you'll see, you know, in the kind of like what's the proposal, the hardware proposal, you'll see, you know, some more there's some ways in which you can actually use CRISPR to get information about the system. Um, uh, so that's the problem of kind of like making it truly closed loop. You know, where you're able to, you know, update it. And one of the things that they also point out is that what you kind of want to do is to also archive particular time points so that you can then do comparisons. So you kind of want to be able to take some of microbes at a particular point in evolution and biobank them, and then use them later to test against, you know, a much later version.
Right.
And then be able to use some sort of metrics in terms of like now these are different, and we see these differences because of these mutations or because of these genetic changes. And we see, you know, these metrics change, right?
Right.
So anyway, it's all covered in the paper.
Yeah.
Or it's
So,
all covered in the write-up, if you will. And it does seem to be something that is certainly out of scope with some of the kind of more inexpensive educational projects.
right.
Whether we can add that without adding significant cost is the question, which you know is super interesting if we can do that. Um, uh, so yeah. Anyway,
Yeah.
that's it's an open repo. I would love to, you know, I haven't. This was like pretty much just yesterday working on
Okay.
this,
Yeah.
and so but I was going to reach out to the Chai Bio guy, because the Chai Bio guy is the guy on Twitter who had got in touch with the OpenEvo guy to say like, hey, what do you know, there is an open source tool. It's been around since 2020.
Yeah.
um, uh, but I'd love to reach out to them. We use the bioreactor project in the cores.
Okay.
And then some of these others, you know, look like they're cool projects, but like their repos haven't been touched in, you know, many years.
Right.
But they've still got a couple. They've still got a couple interesting things that it's like still worth, you know, discussing them as for yeah, design ideas that they have. Yeah.
Yeah, so that's good. Yeah, I just wanted to point out that the LTE, this is where your point about archiving different strains at different generations is actually been quite fruitful in this project because they do
Yep.
archive,
Yeah.
at different generations, and then you can recall that generation, unfreeze it, and grow up that strain, and then you can make
Yeah.
comparisons. So, if I freeze something at 30,000 generations, and then at 50,000 generations, I want to go back and compare to see maybe
Yeah.
something has evolved in the genome or some,
Exactly.
they behave differently on a certain medium. Then you can do
Yeah.
that. And it just makes it more
Yeah.
flexible. Yeah.
Yeah. And I love thinking about this, right? I mean, it's as an undergrad, as a bio undergrad, I kind of learned a lot about this mathematically,
Right.
theoretically, um, and what's really, part of this comes from being around a bunch of founders who care about biomanufacturing. So it's just interesting both to learn about how you can actually do this experimentally to recover all the theoretical results, as well as like what doesn't work, or the actual pitfalls of this, and kind of like what you uh, um, just the pitfalls of it, and problems that actually happen, and like you mentioned, I wouldn't have thought about archiving. um, uh, there's a number of things that I bring up in the paper that came from researching this that look like important concepts to understand for doing this kind of work. But again, really interested in how to make uh, a crisper, accessible research platform that we can kind of like use, um, microbiology to do experiments.
Yeah, that's great. Well, thank you, Morgan. I want to finish up with a few things. So one of the people who has worked in the lab, he's been doing other things. Ankit Grover. He did this. He was doing this European cybersecurity and AI hackathon, and this was something he participated in with some people he knew, and I guess his team won first prize, or they won this championship. So
Nice.
I guess their team was Seaweed Z. I don't know. Let's see if they have. I have two screenshots. I got an update from LinkedIn's that was a little bit kind of sparse. But we won the European Cybersecurity and AI Hackathon Championship Grand Finale. And it says, congratulations to Team Seaweed Z on this fantastic achievement. It's wonderful to hear that the research inspired challenges resonate with you and that the championship led to new friendships and all that. And this is their hackathon leaderboard. So they were on all tasks, they won this championship. And so I don't actually know what their project was. So but just congratulations to Ankit for doing that. So this is well, it's a blue sky post, but it's something that we talked about in the lab several years ago, actually in 2020, I believe, and it's been a problem in machine learning. It's called the double descent problem, or it's double descent theory. It's basically when your error falls and then it goes up and then it falls again. And so this is generalization error versus training error. So generalization error, it falls, it goes up, and then it falls again. It's kind of like this bimodal distribution of generalization error. The training error, however, is unimodal. So the training error you can minimize for training error, but the generalization error is the thing that's problematic here. And so this is generated a lot of interest in terms of how to solve this problem or how to characterize this problem even. And this is a post by Carl Bergstrom, who goes through this problem and this new paper that talks about this in terms of evolvability. So this is in talking about generalization. So this is about generalization error versus training error. So in a model, you train your model, and you try to minimize the error of the training set or the testing set on the training set. And so you can minimize your model, and it's usually asymptotic to 0. And so that's fine. But then generalization is a problem during training error, trying to get your model generalized. And so this is where this double descent problem comes into play. And so they talk about this in terms of evolvability. So there's what I found interesting about this paper was that it talks about evolution and it talks about evolvability. And there are a lot of analogies between evolution and especially simulating evolution and this problem in machine learning where you have this ability to models to generalize. And so this, we can look at the paper in a minute, but I wanted to go through this post first. So this is Carl Bergstrom, and he's an evolutionary biologist, and as well as like a person who does computational biology and things like that. So there are a couple of posts. So the first post is a bit of evolutionary biology. I'm really intrigued by a new perspective piece from Steve Frank that explores connections between how natural selection creates systems that generalize and recent work of machine learning about the surprising capabilities of massively over-parameterized systems. So this double descent is a problem where you improve your generalization and then it kind of degrades. But then if you over-parameterize your model, generalization falls again. So it's this problem of double descent. In other words, the descent occurs under a classical regime, but then it begins to get worse. But then you can improve it again by over-parameterizing your model. And so there is this aspect of sort of the systems nature of this problem. Like, does it generalize? Is that a universal thing in other systems? And in this case, can we observe this in natural selection as well? So this second part here, let's start with the basic machine learning result. When learning about statistics, we all learn about the perils of overfitting. Model a data set with too few parameters, you can't capture the signal, but as the number of parameters approaches the number of data points, you overfit badly. So we generally want to avoid the overparameterized regime, but as you can see, it actually does improve our generalization above and beyond what we can do here with where generalization gets worse as training error falls. So in this region, we might stick with this result where, generalization error drops, but our training error isn't optimal. So we don't want to necessarily go with this region here because we haven't optimized our model for training error, but in a sense, we've kind of optimized it for generalization error, at least to some extent. But then as we keep optimizing our training error, generalization error gets worse. But then when we reach this interpolation threshold where we start to get into an overparameterized regime, we start to get lower generalization error, with lower training error, even though our model is overparameterized. So we do have some problems with going out into that right-hand side of the graph, but that's where we can minimize our generalization error as well. So, and then the question is, why is that? What property of the system is there that leads to that result? In other words, as the number of parameters approaches the number of data points, you perform better on the training data, but tend to perform worse on out-of-sample data. So this is an important point, is that if you over-parameterize your system, you can perform better on the training data. Obviously, you you know the training data implicitly at that point, or you're basically biasing your model towards the training data. But then when you introduce new data or out of distribution data, which means things that the model's not seen before, that it wasn't trained on, then it does very poorly. The surprising recent finding is that if you continue to add additional parameters, things get better again. So there's something about the parameters, the number of parameters, outside of the fact that you're overfitting, that is improving the model in terms of generalization. It's kind of a counterintuitive result. The result is that so-called double descent of generalization error, illustrated in Steve's figure below, shows this problem. The first descent, decrease in error and out of sample data, occurs as you add parameters to the model fit. Error spikes due to overfitting as you continue, but then the trend reverses. But why? The excess dimensions create large subspaces of equivalent training performance by varying generalization ability. So it creates these large subspaces in these large number of dimensions. So you have, as you add your parameters, you add these dimensions, it creates these subspaces of equivalent training performance where you can basically get this result. Within these subspaces, learning dynamics preferentially reach simple, smooth solutions that capture the general structure of the problem rather than its idiosyncratic details. So it's basically a space where you have this sort of generalized model of the problem instead of like very specific aspects of the problem. Note this is a claim about learning dynamics. While there are many solutions that fit the data well, learning dynamics tend to find ones that are smooth and generalize well. Thus, generalization error drops. I don't have a good understanding of why this works and need to read more. I find this very surprising and cool in its own right, but Steve also aims to connect this theory with the evolution by natural selection, which is the part about evolvability. So we have this machine learning model where we have this over-parameterized regime, and so we might translate that to evolution by thinking about over-parameterization as sort of a rugged landscape or a rugged fitness landscape. And so when we have a rugged fitness landscape, we have to be able to evolve our way out of valleys and get our way across the landscape. So if you're familiar with what rugged fitness landscapes are, it's where fitness varies quite a bit across fitness space. And for a population to move to new solutions, it needs to be able to traverse this fitness space. In a very flat or a very smooth fitness space, it's easy to move, it's easy to hill climb up to these optimal points that improve your fitness. But in a rugged landscape, oftentimes there are local valleys that prevent you from like making that smooth move. And so this is very similar to the over-parameterized regime of a machine learning model, where you have a lot of parameters and you have these subspaces where you can do things like evolve new capabilities that can maybe improve your evolvability, ability to evolve, and thus get you to that point. And maybe, if you transfer that population to a smooth landscape, it evolves to that optimal point faster, or climbs the fitness hill faster. So Steve, the author of the paper, observes that evolved biological systems, particularly more complex ones, are good at generalizing about the world. How did they do this? So I kind of gave away the answer. Organisms are often able to function effectively in novel circumstances that their ancestors have never experienced, and this is what we call evolvability. So the idea that you can evolve a solution to a problem that maybe you've never experienced before, but the idea is that you can evolve just in general, just be able to change in a direction that's useful, is something that people, I think, have been very interested in evolutionary theory. And the idea of evolvability comes down to this: not that you're evolving specific traits, but that you actually have these genes that allow you to evolve. So the idea is that there are genes that encourage plasticity. So you might have genes for plasticity. That additional plasticity allows you to change the population to discover new solutions and more novel solutions. And when that happens, you can then have members of the population reach some fitness peak and then drag the rest of the population along with it. That's the idea. And so this, they're trying to build this analogy between evolvability and evolution and evolutionary populations and this sort of generalization ability in machine learning models. So organisms are often able to function effectively in novel circumstances. How is this possible if natural selection has not had a chance to learn about these novel environments? And I just gave it away that there are these evolvability genes, or there's this ability to be more plastic and visit more spaces in that fitness space, more locations in that fitness space. So Steve conjectures that there at least part of the answer lies in regulatory complexity. So this means that we can regulate our genes more effectively, and it gives us a better ability to change. So there's this tuning of genes through regulatory action that allow different states of the gene to be uncovered. And so the high dimensionality of the resulting phenotypic circuits, Steve proposes, enables a sort of generalization that we see on the right-hand side of the double descent diagram that we saw. So the ability to have these phenotypic circuits have this high level of dimensionality translates into being able to explore these rugged landscapes and traverse them to get to a fitness peak. The double descent pattern also occurs if you look at over-parameterized system performance with increased training time. Steve suggests that evolutionarily older circuits that have more evolutionary training, so to speak, are likely to generalize better than recently formed ones. So this is where, where you train these circuits, and the circuits basically consist of genes for evolvability, which are in turn genes that regulate other genes. Like, they might be genes that enable plasticity, or genes that enable the unlocking of different functional aspects of a circuit. And so if you have older circuits, that means that they've evolved longer. That means that they've been optimized longer, or that they have more components that allow for more controlled change in a certain direction or another. So this is interesting because, this involves the study of evolvability. It involves thinking about the parameter space of a model, whether it be a gradient descent model or a fitness landscape. And it thinks about, it makes us think about this, or forces us to think about the universality of this sort of behavior. If you can look across machine learning models and evolution, then maybe it applies to even more systems. So this is the paper here, and so this is the paper here, Stephen A. Frank, who's in an ecology and evolutionary biology program at Irvine. This was published in the journal Evolution, and he explores this problem here where he has this double descent graph and then connects it to evolution. It's a very short paper, but basically it talks about how natural selection encodes learned information in the genome, and so unlocking that learned information is important in terms of survival and increasing the ability of a population to reach some adaptive fitness peak. And then making this analogy with double descent in machine learning. And so specifically increasing regulatory complexity as a parameterization associates with increasing evolvability for the discovery of general solutions. This link between genomic complexity and generalization may have been a primary driving force in evolutionary history. So that is the paper. And then this other paper is a U-turn on double descent, rethinking parameter counting and statistical learning. This is associated with this because it talks about double descent more directly as a statistical learning phenomenon, and then thinking about um, sort of how this phenomena occurs in statistical learning. So in this work, we take a closer look at the evidence surrounding these more classical statistical machine learning methods and challenge the claim that observed cases of double descent truly extend the limits of a traditional U-shaped complexity generalization curve therein. We show that once careful consideration is given to what is being plotted on the x-axes of their double descent plots, it becomes apparent that there are implicitly multiple distinct complexity axes along which the parameter count grows. We demonstrate that the second descent appears exactly and only when and where the transition between these underlying axes occur, and that this location is thus not inherently tied to the interpolation threshold p equals n. And so this points to this idea that, when we think about double descent, are we really thinking about it properly just by looking at this graph? And they kind of spell out what double descent is as a phenomenon. And then, so they say, while most attention has naturally been given to the deep learning setting, double descent is shown to emerge more generally across non-neural models. So known cases include linear regression, trees, and boosting. So in things that aren't neural networks, you see this as well, and then you may see it in evolution. But we have to sort of keep in mind that the double descent model is, you can draw the graph out, but you have to understand where the graph comes from and how it relates to the underlying complexity of the system. So I think that's a nice collection of papers. It kind of lays out that problem, lays out that analogy, and then thinking about like how universal is this, or how do we need to maybe rethink this problem, and how does it all connect?
Yeah, it's something that's well, I think I've mentioned before that this just also reminds me of um, what's called U-shaped behavioral growth in cognitive
Oh yeah,
development.
yeah,
you but uh, yeah, and whenever there's a discussion about the relationship of evolutionary theory with learning, I always think of Richard Watson. um,
oh yeah, yeah,
but it's interesting though, like doing a quick search, I couldn't actually find any um, Watson papers specifically on double descent.
oh
you
wow,
know, which is that's kind of weird. I mean, like as someone who's very much at that, you know, finding the parallels in theory, um, in both theories. Um, you'd think you'd want to tackle that particular
yeah, yeah,
that particular one, just because like so many people make this connection. Um,
yeah,
anyway, but it's a good, excuse me, good topic.
yeah,
And they sorry, and then the other thing I was thinking about was like, you know I mean, we just had a discussion of directed evolution,
right, right,
so like how, I mean, so one, it's interesting again to kind of see that the directed evolution and kind of biomanufacturing industry have some interesting insights in terms of like when you try to make more of something, um, you don't necessarily optimize the thing that you think you're optimizing, and
Right.
and the problems that come from trying to do this without really understanding the agents in the system, so to speak. um, uh, but then there's this wonderful, like, again, in terms of, when you're thinking about synthetic life and or experiments that help us understand the evolution of life, how does adding to the genome get done, and how is that complexity managed, what comes from that, what strategies are kind of found where the more parameters is good. Yeah, again, just trying to think about how to experimentally do some experiments that would capture this phenomena, but with microbes.
Right.
But anyway, yeah, interesting.
Yeah, that's great. Thanks for the comments. All right, I think that was a good meeting. A lot of things we talked about. So I mean, we revisit a lot of this, and I'm sure you'll be revisiting that in your agentic coding exercises and all that. So keep up the good work on that. That looks pretty good. Yeah. All right. All right. That's all for today. See you next week. Thanks. Bye.
Take care.
Take care.